Good morning to my project supervisor, Dr. Nick Norliana, FYP coordinator, Dr. Tinia Idati, and my examiners, Dr. Salmia Tun and Dr. Nur Shakina. I am Girija Shankari, and my final year academic project title is Pigments, Extraction of Dracaena, Angustifolia, and Pandanus Amaryllifolius. First of all, I would like to explain briefly about the two plant species for my research. The first one is Dracaena angustifolia. It is known as Suji plant. It is green in color and rich in chlorophyll content. It is used as traditional medicine to cure gastritis, beriberi, and gonorrhea. The second plant is Pandanus amaryllifolius. It is known as pandan or screw pine plant. It is widely used in cooking due to its aromatic flavor. It contains many different pigments such as chlorophyll, carotenoids, flavonoids, saponins, alkaloids, polyphenols, and tannins. It has laxative and anti-cancer properties as well. So, for my research, I am only going to focus on chlorophyll pigment extraction. Chlorophyll is the green pigment found in green plants. Chlorophyll is important for photosynthesis to convert light energy to chemical energy for the survival of the plants. Chlorophyll pigments are usually extracted to be used for medicinal purposes. It can be used as color additive for food and beverages too. Chlorophyll pigments are natural colorants which can be an alternative to substitute harmful artificial colorants. Alright, chlorophyll can be extracted from plant leaves by many different methods, each having their own pros and cons. The methods are such as boiling, rubbing alcohol, blotting paper, chocolate extraction using different types of solvents such as acetone, methanol, ethanol, diethyl ether, and N,N-dimethylformamide, maceration, and also microwave assisted extraction. Solvents are used for the cell disruption purpose in order to make it easier for the chlorophyll pigment to be extracted out. Due to limitation of lab supplies, microwave assisted extraction could not be conducted. Maceration and chocolate extraction are conventional methods which are suitable for chlorophyll extraction because they retain the quality of the pigments without degrading it. Now, for the problem statement of my research, different plant species have different properties. Thus, different plant species needs different types of solvent to be used for pigment extraction. The best solvent for pigment extraction from Dracaena angustifolia and Pandanus amaryllifolius to produce the highest extraction yield need to be determined. Next, for the objective of my research, first, to examine comparatively the effect of different solvents such as ethyl acetate, ethanol, and N,N-dimethylsulfoxide (DMF) to the output of chlorophyll pigments extracted from Dracaena angustifolia and Pandanus amaryllifolius plant. Besides that, the chlorophyll pigments are differentiated in terms of the color produced and pigment concentration using UV spectrophotometer. My third objective is to determine and analyze the effect of different combination of chlorophyll pigment extracted from both the plant samples. Now, I will proceed with the methodology. Firstly, the materials to be used for my research are 90% ethyl acetate solution, 95% ethanol solution, and N,N-dimethylformamide solution, distilled water. The apparatus to be used are blender, oven, rotary evaporator, beakers and test tubes, distilled water tank, chocolate extractor, and UV spectrophotometer. Next, for the methods, I will start with the sample collection. The Dracaena angustifolia and Pandanus amaryllifolius plants will be obtained from botanists, which is from Mardi. These two plant species can be grown anywhere in Malaysia as long as there is sufficient supply of nutrients and water for its growth and survival. 3 kilograms of leaves from each plant species is used for the study, and they should be collected around 10 a.m. to 4 p.m. Based on guidelines, it is suitable to collect plant sample during this time because the leaves would be dry of moisture. Sharp blades are used to cut the leaf samples from the plant. Besides that, it is crucial to only get healthy and disease-free samples. All the leaf samples will be brought back to lab on the same day to avoid any contamination and unwanted drying of the plant. Next, the plant samples will be washed with tap water to remove any dirt, dust and foreign materials from the surface of the plant samples. This step is very important as a precaution to avoid any bacterial or fungal contamination. The plant samples are air dried and cut into smaller pieces by using a scissors or blade to increase the surface area to ease the drying process. Then, the leaves are left to be dried in an oven overnight at 50 degrees Celsius to remove the moisture content from the leaves. Then, the leaves are to be cleaned and cut into small pieces of about 0.5 cm square. The dried material is then to be homogenized in a grinder for 3 minutes and then sieved with a mesh size of 710 micrometer. The plant samples are pulverized into powder form. Moving on to the extraction of chlorophyll pigments from the powder form of both the plant samples, the total powdered form of the samples are weighed to determine the percentage of moisture content of both the plant samples. Amount of extracted samples and the percentage of every solvent extraction. The dried powder samples of 200 gram is extracted in a chocolate extractor with 90% ethyl acetate, which is 90% ethyl acetate and 10% water at room temperature. The powder samples are placed in porous cellulose thimble and loaded into the condenser. Then, the solvent, which is the ethyl acetate, is added until the powder sample in the thimble was immersed, and the leftover solvent is added in the round flask under the chamber. The heated flask will heat the solvent when evaporated and move through the flow specifically created extraction chamber design. When it exceeds a level set, it overflows and flows back into the boiling flask which contains extracted samples in solvent. The solvents from the combined extracts is evaporated by a vacuum rotary evaporator to obtain pure extract of the plant samples. These steps are done for both Suji leaves and Pandan leaf samples. These steps are repeated for both plant samples using different extraction solvents, which are 95% ethanol and N,N-dimethylformamide. The amount of extracted yield is observed and analyzed to determine the best solvent for chlorophyll extraction from plant leaves. This is done by observing the color difference or intensity of extract when different solvents are used. Besides that, UV-Vis is used to quantify the chlorophyll content in both the plant samples to decide the best solvent for extraction purpose. The wavelength and calculation for the chlorophyll content using the UV-Vis spectrophotometer depending on the type of solvent used is shown in the next slide. This is the equation for each of the for the chlorophyll content used for to determine the chlorophyll content for each solvent used. After the best solvent of extraction is determined qualitatively and analytically, it is then used for further extraction of chlorophyll from fresh leaf samples. The extracts are then combined together using the ratio shown in the following slide. After this step, the color produced from the respective combination will be observed and stated in my research findings. This is the combination of how the two plant samples are combined. For example, one part of Dracaena Angustifolia and one part of Pandanus Amaryllifolius combined, and the color produced will be observed. One part is equals to 20 milliliters. Besides that, the expected outcome from this research is that since the color produced from Suji and Pandan leaves extracts are different, their combination with each other might be able to produce new colors, especially shades of green or maybe a different color. This color difference is important as it can produce new undiscovered colors which can then be used as colorants in the food and beverage industry. Okay, so this is my Gantt chart that I have prepared for my project. I have planned to prepare the raw materials for my research starting from the first week of January 2019 and conduct my laboratory work starting from the fourth week of January 2019 until March 2019. After that, I will continue writing my thesis, chapter 4, 5 and 6 until I have a complete thesis by the end of May 2019. So that's all from me. Thank you.